Thank you for the kind introduction, and thanks for the invitation. And yeah, I'll talk about the work in the lab and focusing on these vision foundation models for microscopy. I'll explain a bit more what we mean by this later on in the talk. Right? First, why do we even care about kind of these deep learning models? Right? We now all say AI about it for microscopy. So what's the motivation to work on this? And I mean, I like to show this slide here, right, with kind of problems that our people, collaboration partners come with us once a week or so, which is something like this, right? So I've imaged the things in microscopy, for example, mitochondria in EM, right? Could be a lot of other things as well. By the way, I think this is from Graham, imaged here at EPFL, this example image. And basically asks, right, something about the morphology in these phenotypes, right? Wanting to figure out if there are differences in the size of the mitochondria between different treatments and so on, right? And so to kind of ask these questions, what we do is quantify this, right? And so the first step that is typically needed there is segmentation, so meaning in this volume EM stack to find all the mitochondria in 3D, and then we can extract kind of the measurements like the volume, the sphericity, and so on we're interested in. This is just kind of a very basic example, but there are a lot of different kind of analysis tasks that can be addressed in this way, right? And so, of course, the most used tools for this now are all based on deep learning, right? And so deep learning basically means that we have this artificial neural network that can learn to transform an input image into the desired output, right? And here we see this for another example in EM, namely segmenting the cell membranes, where we have this input here. This is our desired output, right? Or the cell membranes. And then this here in the middle is the famous UNet, right? One implementation of such an artificial deep neural network that can learn such a segmentation task. Question is, right, if we talk about learning, how does this work, right? So the idea is that we need a lot of annotated data to train these networks, right? So basically we need our EM images, again, could also be light microscopy or any other type of images really. And then we need these annotations or desired outputs, right, that are typically produced by human experts. Like here, all of these painted in EM membranes. And then with this, right, we can train these networks, right? So we have our input, the EM image, we get the prediction from the network, and then we measure sort of the difference between the annotation and this prediction, which we call the loss. And via gradient descent, we use this to update the weights of this network, which you can think about like learnable image filters to then get to our desired outputs, right? So basically to learn this transformation from our input to the boundaries. And so this is kind of really in a nutshell how this deep learning methods work. There are now kind of tools that do this for a lot of different settings, right? Again, focusing here on segmentation tasks in microscopy, right? Ranging from cell and nucleus segmentation light microscopy, where StarDist and CellPose are quite popular. Also the microsam tool from our lab that we'll talk about in a minute, but also mitochondria segmentation, so what we've just seen, MitoNet. And then for many other tasks, right, there are tools that were basically trained this way on already labeled data to solve this. Right? And so this is great. This has led to a lot of advances in image analysis, but there's still quite some limitations here, right? So first of all, there's just many different tools for different imaging modalities, different structures, right? So meaning different segmentation tasks. So just selecting the correct tool, knowing how to apply it takes a lot of expertise. So this is the first limitation. And then kind of inherently in these models, these are quite limited in the generalization. So this means that if they're trained in one image condition and this condition somehow changes, the model will get much worse. So this is an example from our work, right? So this is a network for vesicle segmentation in electron tomography. So and here you see kind of these colorful spheres, right? These are the vesicles. The model actually recognizes them very well here in this tomogram, which is similar to its training data. But then when the tomogram changes, right, so this is from a different microscope in a different synapse, we see that the network misses a lot of the vesicles in here, right? So it finds some, but it does a much worse job. Although, right, as a human, it would be very easy for us, right, to translate from one setting to another. The networks can't really do this, at least when they're trained on a small data set, right, or a data set of only very similar images, so their generalization is limited. And then, right, the third thing is to train a network again for such a task, we need a lot of data, a lot of annotated data. So it's very expensive, time consuming to create this. So all in all, what this means, right, is that the application to new data is still very time consuming unless very similar training data or network trained on very similar data is already available. And so the goal with these vision foundation models is kind of to move beyond these limitations and have a single tool in the end, right, that can address a lot of different tasks without the need for this costly adaptation. And so this is kind of what I want to talk about and focus about mainly today, right, our efforts and the efforts from the whole field to achieve this, right? And so this kind of whole research area can be summarized with this concept of foundation models. And this comes from large language models, right? You all know this by now, right? From ChatGPT, Claude, and so on, the language models that were trained on sort of large parts of the internet. We'll get to how exactly in a second. That can then kind of solve a lot of different tasks when you're giving it different text input prompts, right? So this is just some random example from ChatGPT, but you see here, right, a lot of different kind of inputs, right? From just listing the main function of mitochondria, explaining the difference between electron microscopy modalities, and then telling a short joke about electron microscopy, which is, right, why did the electron microscope break up with the sample? It couldn't handle the resolution issues. You may not find this very funny, but I think at least it's recognizable in the structure as a joke. And what I want to say, kind of with this a bit silly example, is that these networks, right, these LLMs actually do generalize, right? Because there was very likely not an example, right, of an electron microscopy joke in the training data, and you can come up with other examples that were certainly not directly in the training data, but they do kind of actually generalize to these different tasks. So these models, by being trained on very large data sets, actually have a better generalization than what we've just seen with our previous small image models. So the question is, right, can we get similar things now, right, for images, right, a model that actually generalizes to new tasks, to different conditions, and so on. And before we go there, the question is, right, how do these OpenAI or other companies that train LLMs achieve this? Right. So basically, the recipes for building such a chatbot is first having a very large data set, so really kind of scraping a lot of data from the internet, and then training these transformer architectures. Right. So this is basically the latest iteration of these artificial neural networks we've seen on this data, and then also further training it for different what we call downstream tasks, right, which could be things like being able to chat, being able to translate text, being a coding assistant. And so the main part of this training is done in an unsupervised way. So what this just means, right, we have this massive text corpus, we take pieces from it, and then we mask out the end of the text, and the model has to predict, right, the last word basically in this text from the beginning. And then kind of for these specific tasks that come later, right, they are then kind of like more concrete examples, like of good translations or coding that teach these models, right, how to address these specific tasks. Right, so this is in a nutshell, right, how these LLMs, which we also call foundation models, are trained. And of course, right, in the whole deep learning community, there are now many approaches that try to translate this to vision, right? So examples for this are these clip-style models, which connect image and text, right? So this is what you see hinted at here. And so the idea there is basically that these are trained on text and image pairs. We'll also see this in a second. And these then get much more robust in recognizing objects in different representations compared to normal networks, right? So this is basically what you see here. So this is kind of the accuracy of recognizing objects in these images, right? So here, this is just an example of bananas, right? So images where the correct answer is banana. This ImageNet, ResNet is kind of a typically trained normal model, and this is the corresponding clip model. And we see that these are about the same on the actual images, but when we have different kind of representation of the object, right, like these sketches here, these ImageNet pre-trained models really get worse, right? Only 25% are recognized correctly, whereas this clip-style trained models still work quite well. So this is another example, right, of this better generalization by training these larger models with a bit more complex procedures. These clip models are also used now as a basis of text-to-image generation, right? These are all these AI-generated images. All of us have seen, right, kind of models are also underlying it. Then another example of vision foundation models is segment anything. This we'll discuss in a lot of detail, so I talk about this later. And now, of course, also the chatbots like ChatGPT have this vision capability integrated, right? So basically they have something like this clip model also in there that you can copy paste images and ask questions about images in there. Yeah. So how are these models trained, right? So the first idea would be, can we translate this concept of predicting sort of the next word in the text to them, right? That we've just shown about. So what is next word prediction for images? There's this masked autoencoder work that does pretty much this, right? Where the idea is we have our input image and then we mask out a lot of patches in there. The model only gets kind of the patches that are within the mask and then needs to reconstruct basically the rest of the image from this. So this is sort of one of the most popular ways to train these big models, right, in a similar fashion or inspired by this idea of predicting sort of the next words in text. Then this clip model I already mentioned is trained in a bit different way, right, where we have images like here and the corresponding text. And then these are trained to basically put the matching image and text, for example, write a caption from the internet close to each other and non-matching text and images far apart from each other. And so this way, right, this is basically learning kind of a shared vocabulary of images and text. In the case of this clip model trained on 400 million image text pairs. So, right, all of this is great, but gives us sort of interesting capabilities, but none of these, so either this mask pre-training or clip, directly allow us to segment objects in images, which is what we are interested in the context of microscopy. And so now this brings us to the model we'll discuss in most detail, right, which is this segment anything model. So this was developed by Facebook Research. And what the model can do maybe looks quite trivial in the beginning. Basically, it can give you for what we point out in the image, right? You see this cursor here. It predicts the sort of best matching object to the position of the cursor, right? So this is always a blue thing shown underneath where it first predicts a turtle, then predicts a frog here. And then if we put like this red point on top, also manages to separate the frog from the snail on top of it. And so this is what we now call interactive segmentation, where it basically from user input can separate the object from the background in an image. And what is sort of special about this model is that it can do this in a lot of different image settings, right? So these are kind of examples from the paper, which was published in 23, where they showed this, right, from normal sort of street scenes as here to underwater images and so on, right? So we see that this model actually does generalize quite well. And we see even in their initial paper, they tried this on some microscopy. I mean, here this is histopathology, but they also tried some other fairly simple microscopy segmentation tasks and showed that this model generalized to this. So we come to the question, right, how do they achieve this? So there are two main ingredients. So one is that the model architecture is specifically designed for this interactive segmentation tasks. This is what they show here, where the model can take as input different kind of prompts, right? So these are basically these cues like points, also a bounding box that point out which object in the image is meant. And then of course it takes the image as the output, and then it predicts the most likely object for these given inputs. So this is sort of the design of the model. And then it was trained on a very big data set, right? So made up of 1 billion segmented objects in 11 million images, right? And so by having this very large data set that was kind of taken or annotated by the team, so together with this model, or by cloud workers that were hired by the team, it learned basically this generalization, right, to all of these different settings that we see here. Okay, and now this, right, brings us to our work, which, right, when this was published, we became very kind of interested because this now maybe promises, right, to relieve this burden of every time having to train a new model with painfully annotated objects. Rather, we could maybe say, let's use segment anything. Can we solve a lot of these segmentation tasks in microscopy with it? And here you see the very first results that we got when we tried this, right? So these are cells in phase contrast microscopy, something we deal with quite a lot in the lab because we have a lot of groups locally that deal with high content screening experiments. And we see that the model does an okay job, right? So for isolated cells here, it works. So this is basically the green point, right? This is the input to the model. And then the red thing you see is a prediction from it. But as soon as cells get clustered, like here or here, it works quite badly. And then the model also has an automatic segmentation mode where you just put the image into it and it asks it to segment everything in there. And also there we see that it works quite badly, right? So unfortunately, out of the box, right, this model really didn't solve our problems yet, but, and now, right, our works come in it, we could improve it significantly for microscopy, and then see here, right, that it was much better after our improvements. And I'll talk about how in a second, but you can see here now, right, that it can really isolate the individual cells much better based on just one or two inputs, and also that the automatic segmentation you see over here gets much better. So now the question is, right, how did we do this, right? Or mainly, how did my student Anway do this? So first of all, we had to re-implement the training procedure that they used to train this model, because this was not published. The model itself and everything to run it was published, but not the training. And then we added sort of a new part to the model for better automatic segmentation, right? So this we call here the segmentation decoder. And then the main part is we had to actually build a data set that's big enough to cover the diversity of microscopy. And for this, right, we basically went through the literature, through a lot of public databases, and managed to find, right, 30,000 light microscopy images with over 3 million annotated cells and nuclei in there, and something similar for EM with annotated mitochondrial nuclei. And then we retrained the model, right, basically starting from the initial weights, the initial learning from the segment anything model, and retraining it both for this interactive segmentation and for a different kind of automatic segmentation on this data, right? This was now published beginning last year and is already quite widely used. Just to basically show you again that this actually improves a lot over the original SAM model, right? So we see this here, where we have cells in different light microscopy settings. We always see this is the on the left-hand side, the prediction from the original SAM. So the cyan dot here now is the input point from the user. I mean, in our case, we basically just simulate this. The yellow outline is the correct segmentation, and red is what's predicted by the model. And basically for all of these cases, right, we see that our improved model really manages to pick out now individual cells, whereas the original SAM model struggles quite a bit with this. To say, right, there's a lot of cases where even in microscopy, the normal SAM works quite well, right? If cells are more isolated and easier kind of to separate from each other. So there's plugins that just enable using the normal SAM, and this still can be quite helpful. But our model, right, for difficult tasks really does a much better job of isolating individual cells. And this is what we now call interactive segmentation with the model. As I said, right, we also improved it for automatic segmentation. And this is what you see here, right? Basically the outputs from the model for a lot of different microscopy settings, and then just the input without any kind of user prompts. Basically, it's best guess for all the cells or all the objects that's in the images. Yeah. So this now is also quite close to the state of the art for this task. So this is something we just did fairly recently of benchmarking different tools for this. Right, so especially also CellPost SAM, which is something that came out after our model from the CellPost authors, where they also trained a version of their CellPost model based on the SAM architecture. And you can see, right, these comparisons between different models for automatic cell segmentation here. Right, so this is the initial SAM model. This is the latest SAM version, SAM 3. Then this is the prior CellPost version, CellPost 3. And then CellPost SAM, which is sort of the state of the art, but very close in almost all settings to our microsam model. And we have this here, right, for label-free microscopy, fluorescent microscopy, nucleus segmentation also, and also histopathology, where this in our case is kind of a new model that we trained afterwards, where we more or less replicated the effort I just described for microscopy for H&E. So we see, right, that this models that we have are among kind of the state of the art for these automatic tasks, but they enable also this interactive segmentation, right, which is not offered by any of these other tools. So basically, if the model doesn't really work well yet out of the box, you can, with a very few clicks, kind of correct it and get out improved versions, and then use kind of this improved data to also train better versions of this model or of other deep learning models. So it speeds up this process of generating training data a lot, even if the initial model is not fully up to the task yet. Yeah, so as I already hinted at, right, we did something similar for electron microscopy, Here on the left-hand side, you can see the improvements from microsam compared to the original SAM model. And we also benchmarked this against sort of the state of the art at the time and seen quite some improvements that we got. And again, we have this both interactive and automatic segmentation capability within a single model here. We extended the model to 3D in a quite naive way, right? So everything I showed so far was mainly for 2D, but we can also do 3D, where we just process everything slice by slice, right? So basically we start out in the middle and then project these prompts, or in automatic segmentation, we segment each slide individually and then merge the results. So this also works, but it's a bit limited for complex shapes, right? So and I'll come to what we do right now to fix this. And then we used all of this, right, to build basically the microsam tool, which is a Napari plugin that enables all of these features within kind of one tool. Right, you see here, right, this is this interactive segmentation from boxes, also interactive 3D segmentation here again for mitochondria. So this is now available for quite a while already. You can find it under the link here, and is becoming a more and more popular image analysis tool because it has all of these state-of-the-art features for segmentation in one go. So that's the things we did here. For microscopy, just briefly, right, we are of course still working on improving it. So in some follow-up work, we also extended this to medical imaging. To be fair, right, there has been prior work that extended these SAM models to it, and we mainly kind of compared different settings, but found that these training regimes we implemented work better than prior methods. In very recent work, right, we've explored if we can solve other tasks also with this SAM-style models. So namely here to classify already segmented objects, right? So basically, if you have a lot of different cells, they're segmented, you might want to distinguish different cell states, different cell lines, if you have mixtures and so on. Typically, right, you would have to train either like a random forest models that takes simple features, right, like the shape, the intensity for this. So this works great for simple tasks, but hits sort of the limit quite fast. Or you have to train another deep learning model from scratch for this, which can achieve quite high results, high accuracy, but is also sort of requiring a lot of work. And here, what we try instead now is basically to take the intermediate features, so intermediate representations that this model work, extract them for each object that was segmented, right? So this is basically what you see visualized here. And then we train this random forest, right? All of this is inspired by Elastic on top to do the classification. And what we found, sorry, this is quite new, so it's not so obvious from the graph, but we found that basically this works much better than these classical features of just taking the morphology, basically using what the model has learned and then training something small on top, right? So basically with fewer annotations, we can now kind of distinguish different cell types there. And this is something we want to also include into our tools rather soon, right? So that you can not only do segmentation, but then also distinguishing different classes within the cells or other structures that were segmented. And then finally, some work we did a bit earlier is also to try to make this training for new data as efficient as possible. Now, I don't show the plots here, but what we basically found that using only 2 to 5 images in practice is enough, right, for training a new version of the model and really improving it significantly for a given task. Yeah, so all of this was what we did based on the segment anything model. Maybe, right, going through this and of course, me hyping up a bit our work. The question is, are we done with this now, right? Are these segmentation tasks solved? And of course, I mean, the answer is not quite yet, right? I mean, I think we have now very versatile and state-of-the-art for segmentation, but it's always, right, there are trade-offs and still challenges that remain, right? So this kind of fundamental issue, right, of this limited generalization is still there with these models. It's just that their training data is much bigger and diverse than previous models. So in practice, right, it often works out of the box, but kind of still, as soon as you get data that's not really represented in the training set, right, some very more exotic microscopy setting, for example, the model will not really work well. Sort of by design, it's not really suitable for certain morphologies, right? So complex fibrous shapes are difficult for these models, due to our naive solution for 3D, it's also fairly limited for complex 3D shapes. And then these models are just bigger than traditional deep learning models, which also makes them just more heavy in applying them. So it takes longer to segment things, or you need better hardware for this. Then kind of one other thing that's just true in general, right? The segmentation is just one of the tasks that has to be solved, and often the analysis that comes after this, so often it's quite simple, but sometimes it can also be very complex and even more complex than the initial segmentation task. So all just to say, right, there's still challenges remaining. I'll talk in a few slides on how we try to generalize more, but at the current point, right, how we typically operate is that for these challenging problems, we still have to build custom tools or just train custom networks. But almost all the time, we now use, at least if it's a segmentation task, this microsam to generate the training data for this, because this already speeds up the process really a lot. Then, I mean, this is kind of building specific applications for segmentation is kind of also a lot of things what we do in the lab. I don't want to go into too much details there, but just to say, right, that this is what we spend a lot of our time with. So examples here are segmentation of cochlea. These are, right, so structures from the inner ear and light sheet microscopy, right? So this is just quite nice to see, right? This is the whole small cochlear organ of the mouse imaged in light sheet. And then, right, what we do now is being able to segment all the nuclei in here that is then used to sort of validate gene therapies that are tested in these animals. Other examples here in electron tomography to segment sort of the vesicles of synapse, right? So here you see this just a slice from the EM, and this in 3D, all the parts of the synapse that were segmented, and so on. So this is just, right, two examples of these more custom-specific tools that are still necessary, especially as in these cases, if we have large and 3D data. So right, as kind of the outlook, how we want to further overcome, right, some of these limitations is now to extend SAM2, right? So this is basically the next iteration of SAM. So we even have a third iteration that's out there now, but we are still stuck at adapting the second version of it. And so this model can now also deal with videos. And we want to basically use this video capability to also go to volumetric data, basically by treating the time axis as, the z-axis as time. And the idea that we have here is now to build a shared model, right, that can be trained both on 2D images and 3D data to deal out of the box also with more complex 3D shapes. And this is just a sketch, right? But this is basically the idea that we can have something that jointly learns representation for 2D and 3D to solve these segmentation tasks. And the slides are a bit older already, so Andre managed now to set all of this up. So I hope that in like a few weeks, we can actually share the first working prototype of this. So that's all on the segmentation part. So this is really the main thing we've done in the lab so far, right? Just because of this fact that this is often a pressing challenge when applying these two new models. And I'm also happy to take questions before we go to new topics. Yes.
I was wondering about the accuracies that you shared, and I don't know what's good or bad. I see the relative differences, but like 50-ish%, isn't that low?
Yeah, this is actually a good question. It is a bit of let me go to this slide for this, a problem of these metrics. So there's a lot of different ways how to measure this. I mean, fundamentally, they all share a similar idea, right? So basically have the ground truth mask, right? So the annotation actually from a human, and then you check your predicted masks and how well they match. If you try to implement this, there are a couple of sort of specific issues. For example, the annotations are never really fully correct from the human annotators, just because, I mean, there's also ambiguities and so on. So just penalizing these too harsh will lead to very bad values. And what is kind of established in the field now is to do this kind of for different thresholds. So basically saying if they match to 50%, we count this as accurately enough. Then also going to 55 and so on, and then averaging over all of these. And this is still a very kind of strict metric. So it depends a bit on the problem, but typically if we have 50% in there, this is good enough in this metric for most practical applications. And that's why these numbers actually look fairly low, although at least going above like 55, 60% is often good enough. I mean, it's still a pretty important question also because it really depends on the application, right? I mean, there might be cases where if you just need to count or sort of relatively robust, morphometrics, right, like the volume and so on, these numbers are totally fine. But if there's cases, right, where really the exact shape matters, right? Like, for example, there are filaments, there's protrusions from the cell, and you really want to quantify these, then this might not be good enough. So I think this is actually another part of these limitations, right? That actually saying when is this good enough is also not really a solved problem because it's very case specific. Yeah, I think it's a good time for a break. So if there are any more questions, can also continue.
So I was wondering about the cost of retraining some.
Yeah.
So of course you probably retrain from the already trained way.
Yes.
Like how, let's say I wanted to retrain some for completely different type of images. How many images would I need? What would be the dollar cost of this?
Mm-hmm. So I think there's two different parts to this, right? So if you, let's say, first case is you want to train a really good new SAM model that works for a complete new domain, right? So then this is kind of a good, ballpark things, right? We had to collect a really big data set for this. And then the training itself was like on 8 GPUs for a week. So, and 8 data center GPUs. I'm not really sure how to translate this exactly into dollars, but I think this would be if you have it as AWS a few $100s, right? So the training cost is still rather limited compared to the data cost there. If you, again, as I said, just want to do it for a very specific setting, right? Let's say you have your very specific microscopy setting or also any other imaging device, and you only want to have it better for this, then annotating a few images, right? Let's say maybe 50 or so, if you really want to get out the best is enough, right? But then it will only get better, right, for this specific setting, not for like the whole domain. Yeah, and then also the training itself can be done on any standard GPU for this case. Yes, please.
Question like what is the typical part for fiber?
For fibers, I mean, the main problem is that these structures are so small and filamentous, and these models are not very good at details. So this has a bit is related to this architecture design. So basically what they do in the beginning is they cut up the image into small patches and then work on this patch representation, and only at the very end go back to the pixel resolution. So this is specific to these transformer models I talked about, and this just limits their accuracy for very small structures a bit. Right? So this, I mean, it can be overcome, but they are not really built for being very good at fine-grained structures. But there are other deep learning tools that are built exactly for this, right? So it's not like a limitation for the whole field, but for these specific types of models a bit.
So to follow up in this question, 3D case, have you tried to merge your microsum or your microsum 2, which is 3D, with the convolution that you have to?
I mean, this is what is already in there, right? So basically, the segmentation parts is already a CNN. So this does some of this refining again, but the problem is that it starts out from a lower resolution, and one could do even another step and basically say have another network that then works on these masks and tries to refine them. So we haven't done this, but there is quite some work where people basically train these refining style models. Yes, please.
Yes, I think in the original SAM, they have a way of handling what they call ambiguous problems.
Mm-hmm.
So when you click in one place, it doesn't just output one mask, it outputs several masks.
Three, yeah.
Yeah, three that correspond to several objects.
Yeah.
So can microsum do the same?
Yes. Yes. So the model can do the same. We haven't really integrated it into the tool itself because there's some overhead to then do this on the user level, right? But the model itself does the same. In the tool, we just basically take the most likely one, because the model also tells you how confident it is.
So you mentioned that you want to generalize it to 3D and that the original SAM uses the mask autoencoder. Would that work the same for the 3D images? Would you train a mask autoencoder to encode 3D images?
I mean, we would not go back to this step, right? Because we are starting from their trained weights. So basically, we just take what they train for videos, and then we do our 3D training on top. And sort of in detail, it's basically the trunk or the big part of the model. This only works in 2D, also in the SAM2 model. And then there are smaller parts in the end that go over the time, or in our case, the depth dimension. I mean, not here, but it makes sense in general to do this, right? And there's also, we did this in some other work comparing different types of doing this, right? Basically, do you mask randomly in 3D, or do you do like tubular masking and so on? But just because we don't want to redo this, right? And just start from their trained weights. In our case, we wouldn't do this.
Like you showed this that how fast to always like by image.
Mm-hmm. I mean, this of course depends on the hardware you have. If you have a GPU, it's immediate. One image, yeah. I mean, it takes below a second for it on the GPU. On a CPU, it takes quite a bit longer. So yeah, I think around 10 to 20 seconds per image. What we do there in practice is that we pre-compute parts of the model, so that it's then you have to only do this once, and then basically updating these clicks is very fast.
Thanks a lot. I was wondering whether you could comment on how you, what experience you have with, or kind of you made with turning this into automated segmentation, and how big of a challenge it is to go kind of from this promptable segmentation to automated segmentation.
Mm-hmm. So what we do there is that we basically train a new part of the model, right? So new decoder that branches off this big part of it, and then this is kind of a bit separate from the interactive segmentation, right? And we do something more or less similar to what Salpos does in there. Basically, we predict foreground, background, and then certain distances, and use a watershed for the segmentation. Because we found that what Sam did in the beginning, which is covering the image with a lot of different prompts, was just very inefficient, and kind of this more dedicated solution worked better than in the end.
And maybe as a follow-up, do you see there's a kind of much improvement, kind of possibility in there? Kind of, is that a big part of the problem? Kind of the different methods you use for turning semantic into instant segmentation?
Yeah, I mean, there's definitely room for work and improvement there. So basically, how the main thing is how to best find representations to be used. And we're also still looking a bit into this, especially now with the 3D. We are kind of looking into this again to see how to do this well for anisotropic 3D data. Right. So yeah, there's definitely room for improvements there, I think. And I mean, I hope we can share this soon, what we do in the 3D case, which is, I hope, now a solution that actually works well, but we still need to benchmark this a bit.
Cool, thanks a lot.
Thanks for all the questions. It's very great. I'm also, the rest will be just quick overview over some other work, right? Because segmentation is not the only thing we work on in the lab, right? So the other thing is related to this representation learning, which is basically the question now that we've solved, or more or less, or at least made advances to the segmentation task, right? So we can basically easily go from this image to whatever segmentation we want. And hopefully in a few years, right, this will really be routine with the models we and other are developing, what comes next, right? And I think, because we are not doing this segmentation as an end in itself, but we really want to do this to either measure specific things, as I shown in the beginning, but often also to discover, right, new things, right? So for example, if we have like, sort of some treatments, we want to look at how this changes morphology, right? We often don't really have a certain or really clear hypothesis and so on. And so the question here in this representation learning is, can we also use these models to help us with this, right? Because the classical way of trying to basically enumerate all the possible morphologies, finding ways to quantify them, and then comparing them can be, also a lot of work and quite limited. And so this idea, right, is really coming a bit from drug discovery, or at least this drug discovery is a field in microscopy that uses this most is to try to basically learn representations of a cell with a network that are more expressive than the classical morphological descriptors like size, texture, and other kinds of morphology. And so this is here from work from sort of and Carpenter and related groups, where they basically show this for these cell painting assays, that training a network in an unsupervised, or in this case, in a weakly supervised way, that basically just means they see the kind of cell painting assay and just sort of some kind of quite general information about the compound where it was used, actually can work better for sort of the task they're interested in, which is in this case the mode of action prediction, than just the morphological features. And of course, right, and Carpenter's lab developed cell profiler, so they're also really leading in these kind of morphology. Yeah, and so this is basically the idea here, right? The way this works in a tiny bit or more detail is basically that, right, we take the images, we segment all the cells there, and then, right, we have like this network, again, similar neural network to what we've seen before, that then predicts for each cell, right, kind of the condition, the perturbation, and then if we apply it to something new, we also take these features and then we use them, right, for the specific task we're interested in, like the mode of action prediction. So this is kind of the rough idea there, and there's a lot of different ways how this can be implemented. I think this is sort of, yeah, the hope, right, that this can help us also with other tasks that come after segmentation. So this is an example from the lab, right, where we try to use this now is kind of also tasks in personalized medicine, where we have a group at the University Medical Group that have these patient-derived organoids, right? So this is of pancreatic or colorectal cancer patients, and then they use high-content microscopy, right, to basically compare different drug screens for these organoids, and right, they get of these bright field images in here. And basically what's quite easy to measure there is do the cells grow or not? Do the organoids grow or not after treatment? But the sort of hope and hypothesis is that there are more subtle morphological features that could be used and that could, hopefully in the end, be used kind of for the treatment recommendation, or at least some stratification of the patients. This is all quite, yeah, in the beginning a bit, but we are basically following the same or similar approach as I just outlined there for the cell profiling, right, where we segment all of these individual organoids, and then we have a network like this here, right, that is trained in a more unsupervised way, and there we then try basically to correlate these representations learned with the patient's sort of genomics data. And all of this is still a bit in the beginning. There's definitely a lot of kind of challenges here, right? So what we would really want for these cases is also interpretability, right? So basically once these networks give us these representations, it's great if we can sort of classify things of this, but what would be really important for these cases is to actually have like some explanation, basically in this case, which morphology is actually predictive, and link this back then to something more mechanistic, right? Basically sort of link it back to a pathway or so on. And in general, right, all of these things are more exploratory, which makes validation and so on quite more challenging. But I think, right, this is sort of in the coming years something that's quite promising, and also hopefully can be connected to other data, right, like omics and so on, in a better, more fundamental way. So this is kind of the other sort of research direction that we're starting in the lab. And then finally, just one slide, right? The other thing is also looking at things that are smaller than cells, right? So basically either particles or proteins in cryo-electron tomography, right? So it's just a sample image you see here, but where we try to also solve this task of identifying proteins in cryo-ET with deep learning to hopefully get better than the traditional template matching approaches, and then also looking at super resolution microscopy, right? So this is an example where we have a combination of expansion microscopy and super resolution microscopy. So this is from the Risoli lab in Göttingen. And then we build sort of these methods to reconstruct the protein shapes from this, basically doing something similar to cryo-EM, but with light microscopy. And now also very recently, we started to look at analyzing sort of biomolecules and their dynamics with MinStat. But yeah, this is just to say, right, kind of this is, I think, another next frontier. And of course, a lot of labs working on this already. Once these segmentation tasks are more in control, to really look more detailed into the molecular landscape with these tools. yeah, just right, briefly in conclusion, right, so the focus were really these models for segmentation or other image analysis tasks, right, like classification, also tracking, which I didn't talk much about, but which are also now being integrated into these tools, where right, these foundation models already kind of minimize the need for retraining, so make all of this access to advanced solutions easier, and I think will really improve in the last years, right, to give us more universal solutions for self-segmentation and similar tasks, right? And then some of the next kind of frontiers there is this representation learning, which I'm personally very excited about. Basically, once we have segmentation more under control, actually being able to do something with this. And then, right, the last thing I just hinted at, right, the visual proteomics to basically look at protein shapes, protein dynamics, and interaction also through microscopy. Yeah, but just briefly a shout out, right? I think this was announced and some of you have signed up. There's also the workshop tomorrow. So there we will really focus on this microsam tool and yeah, have some hands-on experience on how to do it, how to use it for example data or your data. So for whoever from the workshop is here, more on this tomorrow, and you can already check out some of the materials here. And then of course, a lot of thanks for people supporting this work in Göttingen, to my group, right? So the students, PhDs, and postdocs who actually implemented everything I showed today, and then also the funders that made all of this possible. And this is, thanks a lot, and I'm happy to take any more questions.